It's my great pleasure now to introduce our second speaker for this afternoon, which is Dr. Constantin Aliferis. Dr. Aliferis, as you know, is a professor of medicine, chief informatics research officer, and director of the Institute for Health Informatics at the University of Minnesota. He was one of the founding directors of Vanderbilt's training program in biomedical informatics and founding director of the New York University Center for Health Informatics and Bioinformatics. He has numerous years of training, over 17 years of formal graduate and postgraduate training in medicine, computer science, bioinformatics, which makes him extremely qualified to speak about his topic today. Dr. Aliferis is a well-funded research program that is focused on high-dimensional modeling and analysis designed to transform biomedical big data into novel, actionable scientific knowledge, very apropos to today's topic. So please welcome Dr. Constantin Aliferis.
So thank you very much for the introduction and also for the opportunity to participate and also to be a speaker in this very timely and very vibrant conference. My personal research has to do with high-dimensional data analytics for molecular profiling, for clinical risk prediction, and also for reverse engineering of pathways for mechanistic discovery. My administrative leadership roles at as director of the Institute for Health Informatics and chief research informatics officer are to participate and lead the creation of infrastructure, computational and informatics infrastructure, facilitating, among other things, the goals of personalized and precision medicine. Now we're going to change the pace a little bit, and we're going to talk about data science and informatics. I hope that it will become evident why this is pertinent for this conference. I hope to be able to show you that data science techniques, scientific methodologies and technology, as well as informatics, are absolutely essential in the team effort that's involved in realizing the vision of precision medicine. As a matter of fact, in this presentation, I will refer back to several of the questions that members of the audience and speakers addressed previously in terms of doing both research and delivery, standards necessary for data storage, and also, very importantly, for deployment in a clinical context. We will switch levels from high level to lower level as it goes. So there has been a tremendous set of successes in our ability to deal with the challenges of precision medicine. Many of them come from the domain of computer science, information science. If we open a little bit the scope, because today we've been talking primarily about mostly reactive and single gene-based precision medicine, I would like to really examine a little bit the broader scope of precision medicine and to just discuss very briefly the various varieties of workflows, research processes, use cases, and so forth for the most major type, the most important types of precision medicine as we know it today. So first of all, precision medicine is not very new. As a matter of fact, if you think about clinical risk prediction, staging, models that deal with assigning patients to various treatment groups on the basis of clinical data markers alone, not pharmacogenes, this has been going on for several decades. And this is a very, very important part of established as well as future precision medicine efforts, especially as the scope of data to be analyzed, the complexity and sophistication of models, the life cycles of this modeling and deployment gets shorter and shorter, and the predictivity gets higher and higher. Another very important form of precision medicine has to do with complex panels and complex patterns of either genetic or genomic and various other types of omic variants and variables that can be recorded and then can be modeled and often linked with clinical data and imaging as well to create complicated decision support models that either confirm diagnosis or provide prediction of prognosis in the natural course of disease or under various therapeutic conditions in response to therapy. And we have some excellent examples of established and very successful subtyping and disease precision medicine modalities. Oncotype DX is a primary example of this type of precision medicine that has a very, very important characteristic, which is basically it's not very human interpretable. So these kinds of models are very complicated and often act as black boxes. They're not as open to clinical on-demand interpretation as single-gene precision medicine is. Excuse me. Sequencing tumors, obviously, and related oncology guideline, as well as last resort type of attempts to help patients that do not respond to frontline treatments, is another type of precision medicine that's of extreme importance that's only growing. Of course, several speakers mentioned preemptive pharmacogenomics. We're going to talk about this quite a bit in a few minutes. And of course, there are two types of preemptive pharmacogenomics. One is the kind that we observe today in leading centers, a small subset of existing hospitals and other medical centers around the country, where patients are being genotyped upon admission typically, either with a restricted panel, as has been the case so far, or with expanded whole genome and whole exome sequencing, as it is the recent trend. But there is also the future version of preemptive pharmacogenomics, where everybody's genome has been sequenced at birth and then follows them for life in their medical record. And we haven't seen that so far because we don't have a genomic medical record or other infrastructure and frameworks that are needed for this. Of course, rational treatment development is part of precision medicine. And lately, we also see direct-to-consumer sequencing, pharmacogenomics, or lifestyle-oriented modalities and sensors typically delivered through smart devices, wearables, smartphones, and so forth. So in other words, the universe of precision medicine is very expanded, and single-gene pharmacogenomics is a very, very important component of this very big milieu. However, we need to, I think, to be thinking in terms of the broader terms as well, because this ecosystem is being highly interactive and evolving, and everything happens in context. So what's the role of data science and informatics in all of this? Well, first of all, most precision medicine efforts and infrastructures have big data characteristics, and that's a very important element in conceptualizing them and dealing with them on a practical level. So data size, volume, used to not be a problem. Moore's law, which practically held true for around 40 years, was saying that every year and a half, the size of chips and their processing powers of CPUs would double. And therefore, production of new data and so forth would not be a problem because we would be able to keep up with data and so forth. And that was true, but until 2010 or 2012 or so, this stopped to be the case. And now we're lagging behind. Similar laws for disk space and bandwidth also kept pace with the data volume for some time, but now no longer keep pace with them. And currently we're dealing with extreme volumes of data from the gigabyte range, which is roughly the scale of storing one full human genome, to petabytes, for example, which is 1,000 gigabytes. For storing the 1 million genome project, we need 1 exabyte, which is 1,000 petabytes, if we account for intermediate results. And then we are at the point that we're talking about zettabytes, which is basically 1,000 exabytes when we're talking about global data storage. So as you can see, we are faced with extreme volume of data that's being produced and needs to be analyzed and brought into context. Now, in the health sciences domain, let me give you some examples of very big data sets like this that are highly pertinent to precision medicine. The Veteran Affairs Million Veteran Program, which aims to collect data from DNA sequencing of 1 million veterans and link them to the Veteran Administration's medical record system, is one such example. The Precision Medicine Cohort Initiative, which is of exactly similar scale, is another such example. And then you have other various international initiatives, such as the UK Biobank and the Chinese version of that, as well as local big healthcare systems initiatives of that sort, like the Kaiser Permanente research program on genes, environment, and health, which are producing massive, massive data sets of that sort. And now the definition of volume that makes a big data set in healthcare is log n times p bigger than 7. What that does mean? That if you take the number of patients and multiply with the number of data elements, say variables, to be observed, and you take the decimal logarithm of this, if it's larger than 7, it's a big data data set. So for example, if you have 1 million patients, which is 10 to the 6th, and you observe 10,000 variable codes, for example, like disease codes in a sparse matrix for them, along with other data elements over time, that is a log n times p of at least 10. So it's a big data set. But the volume is not just the only element of big data. It's also the velocity, which means the rate at which data is being produced and transmitted, the variety, which means the variability in the types of data that are being described. And medicine and health sciences are notorious for their extreme variety. Think about it, that we treat thousands of different diseases, and many of them have various disease subtypes with thousands of different medications and procedures, performing thousands of different tests over time. So I don't think that there's any other industry that has higher variety than, say, the healthcare industry. And these are the traditional 3 V's of big data, but there are 2 additional ones that have been added recently, which is the validity of the data and the value of the data. And this plot here shows how publications in big data and health sciences have started to explode around 2010 or so. Now, these plots show how the various components of health sciences, which are medical, specialty data, omics, data of various sorts, and public health data, how they're contributing and how they compare with one another in terms of their volume. So this plot, for example, and actually, I'm sorry, I just realized that my pointer does not translate to the screen. Maybe this will do. There is a small skinny one. Okay, I got it, thank you. So this plot here, for example, shows that clinical data dominates in the log 5 to log 10 region for number of variables. And then there's also some huge data sets here in the extreme volume range for data elements, with the other types of data being very similar except for clinical in between. This plot here shows the log scale of number of subjects. And as we can see, they're highly comparable in the upper range of big data here among the three types of data sets in health sciences. And you can see also in later years how the scale of combined number of data elements and patients has been exploding above the big data thresholds. Now, sequencing is definitely a very, very big component, as we saw, in terms of participation in the omics part of big data. And part of this is cost. In 2007, the cost, of course, before 2 decades or so, the cost of sequencing one human genome was in the billions. Around 2007 was in the $1 million range. Recently, it is in the $1,000 range. And that is translated into explosive growth that will only gonna get better depending on various estimates. And informatics and data science are really participating hugely in this because via a variety of data generation, computer processes, and also the corresponding files, they contribute directly to the creation of these huge data sets. So what are some of the technologies that have been developed to support managing all of these huge and very complicated big data sets in health sciences? One set of technologies has to do with parallel and scalable computing. And cluster computing, which means you put together a lot of low-grade CPUs and into big farms of computers in order to do computing. Parallel computing has been around for a long time, but has been commoditized recently, and we have very strong, very powerful computing environments that are based on these architectures. Grid computing is another variety where systems can put together temporarily borrowed computing cycles from participants, typically volunteers who want to participate in scientific projects, and this has happened a lot in astronomy and health sciences and elsewhere. Another very, very important development is utility computing, and you have heard this in the form of probably cloud computing or elastic computing, same thing. This means that instead of having built a very, very big supercomputer, we can now go as scientific institutions or even individual investigators into a cloud computing provider and use as much computing power as we need at any given point without setting up this infrastructure and paying only for what we use when we need it. And the ability to do that is very closely related to virtualization, which means that one computer system can emulate another, and therefore in this cloud computing, you can set up many, many instances of other computer systems that are virtually supported by this cloud environment. And accordingly, there are things like infrastructure as a service, platform as a service, and software as a service, depending on exactly what's provided to the end user versus what the end user has to build. So if everything is provided ready, including the applications, this is software as a service. If only the low-level computing infrastructure is provided, and then the virtualization, the frameworks, and the applications have to be provided by the users, this is infrastructure as a service. And in between, we have platform as a service where the user provides only the applications. Recent developments also include so-called shared nothing architectures. Famous example of that is Hadoop and its components, the Google file system and MapReduce, which allow for very, very high scalability of processing. They allow for commodity hardware to build very powerful supercomputers. They allow for computation to be pushed to individual nodes, enabling thus extreme scalability, low cost, redundancy, and very low setup costs. So a lot of progress has been made in the last years in highly scalable computing architecture. There's still many challenges, of course. Some of this is costly in terms of personnel. It involves complex software development. It's not as interoperable. There is support stability problems. Another problem not mentioned in this slide is that when you want to move, for example, huge sequencing data to a cloud provider, the rate limiting step is this transference of data. It may take weeks or months for very, very large data sets. And also HIPAA compliance is another health sciences related issue. And there has been some progress, but we don't have a fully compliant, strictly speaking, cloud solution yet. Now, the ability to generate data, store it, transfer it, etc., is one big part of data science, but the analysis and the modeling of data to generate new hypotheses or to test specific hypotheses is another very, very, very important aspect. So very, very briefly, because we don't have time to go over details, I'll say that there has been several decades worth of advanced analytics developments that have converged to the last decade or so to our ability to now analyze data sets with millions of variables and a few hundred patients and make sense of them without overfitting the data, without producing noise, without producing irreproducible models. And such analytics involve, of course, a whole host of molecular profiling analytics and subtyping, causal graphs for genetics analysis and genomics analysis and pathway reverse engineering, very complex protocols that calculate predictivity and other future errors of models from one data set, multi-level and multi-layer systems and network science approaches to model data, and a whole bunch of others. So let's go back to pharmacogenomics after we saw a little bit the big picture and mentioned a number of very important technologies. There are three important dimensions in pharmacogenomics that relate to all of that. One is discovery, the second is validation, and the third is deployment. And discovery has to do with proof of concept, establishment of associations between genes and variants with clinical phenotypes such as outcomes and disease and so forth. Another aspect is validation and in terms of reproducibility in different cohorts and so on, in different environments, but also economic cost-benefit analysis that says whether something is practically feasible or not. And the third one is deployment. In other words, okay, we know that there's a pharmacogene that predisposes to a particular clinical outcome, response to treatment, ability to activate a drug or metabolize it and so on, but how can we deliver this in real clinical practice? So in addressing those matters, let's first observe that discovery and deployment are really very tightly linked together. This diagram here shows that in a prototypical setting where someone is doing patient genotyping, it's entirely possible and in most realistic circumstances part of the workflow to filter variant information into actionable and non-actionable or unknown, and then to be able to perform on one hand clinical decision support with the known actionable variants, but also to be able to perform discovery with the unknown variants and validation and so forth for the ones that are in lower levels of evidence support. I think Dr. Jacobson presented the same graph previously, which shows how much relevant is the current knowledge of pharmacogenes to clinical practice, but also it shows how much more potential there exists. This big part here shows the discovery potential that we have in our hands, not just the delivery of this known slice here, but how much remains to be discovered. It was also discussed previously, what are the barriers? It's very interesting that in this discussion we keep cross-referencing with one another's talks, actually. It means that there is a lot of very important core publications and studies and so forth underlying our discussions and scientific thinking. The only thing I would like to add to this, which was discussed previously by another speaker, is that one obstacle for actual delivery is not just the individual components, but also a systems-level approach for a healthcare system to have coordinated action and enterprise strategies for delivering pharmacogenomics and other precision medicine. It's not enough to say, oh, I have this component, that component, the other component. Unless these are all brought together, it's not going to work. Excuse me. So a very, very powerful concept is that if someone links biobank specimens and the ability to run genotypic assays on those specimens, and of course having the legal right and consent, patient consent and so forth, or human subject consent to do that, combine this with the electronic medical record, this can become a very, very powerful engine for discovery. Now, of course, there are some very tricky elements in this effort. What efforts such as the eMERGE network and others have shown is that a very tricky aspect that has to do with electronic health record data is the careful and reproducible and transferable definition of clinical phenotypes. So because the health record is not designed for research, but it is designed for clinical care and is a transactional system, which means it looks at specific encounters as the core window of observation, it suffers from deficiencies relative to well-designed, for example, GWAS. Moreover, all the observed clinical elements in an electronic health record suffer from extreme selection biases. The vast majority of data elements are not observed randomly or all of them, but they're selected based on what the care provider has observed on the patient previously. So it's a sequential decision-making process about what to measure, how to intervene, and this keeps going until the patient's problem is resolved or nothing else can be done. So from the informatics standpoint, in order to make this discovery engine possible, several standards for data storage, transmission, and reporting are absolutely necessary, and they have been developed for both front-loaded and late mapping of observations to data elements, to canonical codes and names, including RefSec, the Human Gene Nomenclature Committee, the Human Genome Variation Society, LOINC, HL7. Someone asked about HL7 previously. The answer is version 2 of HL7 now has standards described to allow for the description and transmission of genetic tests, whole genome information, and patient reporting. So you see the work that's being done on the standard side of things is really keeping up, and actually it may be a little bit more proactive and forward-looking than our ability to deliver the clinical advice at the bedside. Also, there is a number of external references and symbols in a variety of databases where this information is being deposited, including, of course, the online Mendelian inheritance in man, PubMed, PharmGx, clinicaltrials.gov, and others. Now, the model of FIWAS was described previously briefly, so let me make some additional comments if I may. So in GWAS, we have several genotypic variant information. And when I say several, I'm talking about in the range of a million or above, say 1 million SNPs. And then there is the correlate, the response variable, which is a singular clinical phenotype, typically a disease, or in some cases, treatment response. In FIWAS, what we have is a smaller set, traditionally, of genetic variants, typically some panel of SNPs, and then a very large number of clinical phenotypes, typically disease. And the same process that we use for univariate association testing in GWAS is applied in FIWAS. Now, and there are variants also because you can try for different types of phenotypes, not just disease, but could be a number of traits, clinical outcomes, and whatnot. Now, these two models, although the naive approach to say, oh, they're the same, they're inverted but the same, they're very, very different. They are very, very different. Why? Because if you do not do full genotyping, but you do a restricted genotype, so instead, let's say instead of doing 1 million SNPs, you do 100 SNPs, just to make a whole round number example. Then your prior of hitting the true genetic cause of any particular disease is 10,000 times smaller than with a traditional GWAS. In order to overcome statistically this prior, your odds ratio has to be astronomical. Your sample size in a FIWAS is not designed for any particular effect. It is what it is in the medical record, which means that for common diseases, it's going to be high. For uncommon diseases, which are the majority, it's going to be extremely low. As the ability to do whole genome sequence and whole exome sequence becomes a reality, the first part of the small priors will be overcome. The other part still remains a problem. I'll go back to the slides. Other problems that FIWAS have that without site aggregation of data into a bigger data set, the power is very small, but in order to aggregate this data, it's very difficult. And it's very difficult because of the data element differences between sites. The advantages, though, of electronic health record FIWAS style-based discovery is that you can reuse patients across studies. The cost of a FIWAS versus a GWAS is tremendously attractive. So we're looking at 20 times less cost for a FIWAS versus a GWAS. And the speed of recruitment has been estimated to be about 10 times faster in a FIWAS than in a GWAS. And also it has the potential for pleiotropic effect discovery, meaning not discover just one effect of a particular genotype, but multiple effects, if such multiple effects indeed exist. Now, a critical component in making this a success is the ability to compute reproducible, reliable phenotypes of disease, outcome, or other from the medical record. And that's not a solved problem. This is a work in progress, and there are various approaches how to do this, using ICD-9 or ICD-10 codes, using aggregations of those, which are called FIWAS codes, or using very complicated protocols for computing phenotypes from electronic health records, which are known as computable phenotypes. And actually, there is a repository of such tested and validated computable phenotypes in this web resource here. And of course, there are other technical and methodological problems. For example, when someone wants to discover pleiotropic effects, there are a whole lot of causal models that may be happening, generating the data that will not produce a good false positive to false negative ratio in the electronic health record FIWAS discovery setting, and they require different techniques other than univariate association hypothesis testing. Other technologies and methodologies that are being used to facilitate discovery have to do with the mining of the literature. This plot here shows how the pharmacogenomics literature has exploded in recent years. In order to catch up with that literature, automated means are needed, and indeed, there is excellent work that's happening in the domain of natural language processing of such literature to identify what articles are talking about what particular pharmacogenes, to extract relationships between genes and variants and phenotypes, and so forth. The evidence-based processing of pharmacogenomics is definitely an important work in progress. I'm not going to go into how CPIC and PharmDB manage this, because this is probably known to everybody. And let me also mention very quickly the following elements. We don't currently have a genomic health record, meaning a fully integrated health record that includes genomic and genetic information over time, and the decisions made about the patient on this basis, and decision-making aids proposed on this basis. There are various technical desiderata proposed by informed people about this, but we don't have an implemented version of that. However, the current model is to have an omics ancillary system, something that sits outside the health record and communicates with the health record via either naive protocols such as sending PDF reports, or via more sophisticated communication channels and protocols. This is work in progress. There are groups in the eMERGE network in particular that are making very good progress in evaluating these various approaches. And finally, the cost estimates of pharmacogenomics are really interesting, and what are the various components. As a reflection of how big progress has happened in informatics, data science, and computation, the costs are extremely low in that regard. They are very, very high in identifying phenotypes, because it's basically a manual endeavor so far. Once we have truly computable phenotypes, this is going to go much lower. So in conclusion, obviously, pharmacogenomics is an essential component in the personalized and precision medicine universe. Discovery and deployment are very tightly linked together. We have been observing, as a scientific community, many advances in addressing many of the key elements in both discovery procedures and delivery. However, a lot of open problems exist. And also, pharmacogenomics has many characteristics of big data, both for data generation, transmission, management, and the analysis viewpoints. But as I said, there have been significant breakthroughs that address those. Precision medicine is a team endeavor. It's not a single discipline. I don't think that there's going to be a single clinical specialty that will ever allow the expression precision medicine. It's going to be a team effort. One key participant in this team is data science and computer science and informatics, biobanking, medical records, and I hope that I gave a little bit of an outline of how they relate. I'll be very, very happy to answer any questions you have during the panel discussion. Thank you.